precious for that, just teeny tiny little bit, but quite important for mitochondria called cardiolipin, and also its derivative of the gas and how you control mitochondrial dynamics and its quality. Before that, I haven't had a chance to visit Cancun, but I would like to share with you this beautiful, iconic scene in our campus, National Taiwan University, is the Royal Palm Boulevard. So I would like to invite you, if you have a chance to visit Taiwan, I'll be more than happy to be your tour guide. Okay. So please join me for take 5 seconds to breathe. Inhale and exhale. And the next question is, what do you breathe for? I don't know theoretically who you are breathing for, and you working so hard every day, but technically we breathe for our mitochondria, because the oxygen that we inhale every 5 seconds is actually working as an electron acceptor, that during the electron transport, the mitochondria need to use oxygen to convert the free radical into something they can deal with. And mitochondria is actually an endosymbiotic organelle in our cell that actually used to be a parasite that internalized into our ancient nucleic cell and then become part of our cell. And due to this kind of evolution origin, mitochondria first have two membranes, double membranes. And outer membrane are more similar to the bowel membrane of our other membrane structure, plasma membrane, nuclear envelope, or ER. But the inner membrane is actually more like bacteria. And then they have a very unique structure we call cristae that is enriched with the electron transport enzyme that is responsible for ATP degradation. And due to the nuclear power plant nature, that you can imagine it's deal with a lot of damaging agents like free radicals. And they also suffer from the risk of protein and lipid damage by those reactive oxygen species. And therefore, the mitochondria need to do a lot of work to prevent the damaged mitochondria to become a major population in our cell. So they need to cleverly to get rid of the damaged part through the fission process and also through the fusion process to maintain their good amount of mitochondria for their function. So this kind of dynamic and turnover is very critical for mitochondria to maintain our cell ability and activity. And many proteins, including the DRP1, mitochondrial 1, 2, or OPA1, those vitamin-related proteins have been reported and well studied for their critical function and role in control the dynamic and the quality of mitochondria. However, there is a hidden role and also very critical role is actually the lipid. That is actually one of the major components in the membrane of mitochondria. And similar to most of the other membranes in our cell, that phospholipid is also the major component of our mitochondrial membrane. But there is one particular lipid called cardiolipin is only enriched and synthesized in the inner membrane of mitochondria. And it also has a unique structure. First is different from the glycosylphosphate we saw from the previous slide that only have 2 S chains. Cardiolipin actually have 4 S chains. And with this very unique cone-shaped structure, cardiolipin is perfect for the mitochondrial inner membrane cristae formation. It's a very diverse S chain. And with this unique structure, the cardiolipin is critical for the membrane architecture, protein import, and function of those electron transport chains. However, it also contributes to the dynamic mitology and also apoptosis of mitochondria. How come? This is because when mitochondria are under stress or injury, it will externalize from the inner membrane of mitochondria all the way to the outer limit of mitochondria. And therefore, outer mitochondria coupling will trigger either apoptosis to induce the calcium export forming the BDAC pore, or promote the fission through binding to BRP1, or eventually promote mitology because cardiolipin can bind directly to LP3 to trigger mitology. So this is a multiple function cardiac dysfunction. However, it doesn't happen without cost. Actually, for the cardiolipin, it's passed in the inner membrane of mitochondria to all the way to the outer membrane of mitochondria. Ultimately, it actually needs to be transferred and converted all the way to multiple steps. But I will skip this part because it's not the point today. So what the point today is that the destination for the cardiolipin to the outer membrane of mitochondria is not the end of the story. It actually can have some turnover at the outer membrane. Back to 2006, in a microformer, they discovered an enzyme called phospholipase C of mitochondria. At that time they called mito-PLD, but later it was renamed to PLD6. This enzyme is very particular or specular in the cell because different from other PLD family members, this protein first had mitochondrial localizing, and then actually localized to the outer limit of the mitochondrial membrane. And then form dimer was this active site facing outward. And importantly, the function of this enzyme is to convert or hydrolyze cardiolipin into phosphatidic acid, another phenyllipid in our cell. And so that's what they proposed that time. And the function for this enzyme is to actually promote mitochondrial fusion. So this is actually opted from that we know originally of the cardiolipin to promote fission and mitology. However, with this, some have been speculated is converting enzyme of the PLD6 that can control dynamic of mitochondria. However, what is missing is what is the factor for this kind of conversion that can promote mitochondrial fusion rather than fission. And this is the final result. I would like to also try to convince you that we find that the missing piece of the puzzle is underneath 3. That is a protein that can bind directly to phosphoric acid, and due to its hexameric nature, it can function as a tethering protein to tether two close position mitochondria to promote fusion. Okay, so this is a story I'd like to share with you. Okay, before I will go through the data, I'd like to shortly introduce you what is NMD3. NMD3 is actually one member of this superfamily called nucleoside diphosphatides. Their traditional function is actually to catalyze NTP to NTP by utilizing the phosphate group from ATP. And for this member of this superfamily, but many of them have been reported to function as the fueling enzyme to provide GTP for those GTP8s to function in membrane trafficking. However, for the NMD3, it actually have a very unique study identified that the function of energy is actually critical for mitochondria. That the human patient with amylase mutation would develop neurodegenerative disorder, and skin fibroblasts from the patient actually show abnormal mitochondria. Compared with wild-type control, mitochondria become more fragmented. And this is because of the function of the amylase, but not through its kinase-dependent function. I already mentioned that this is a GTP-producing enzyme, right? So it transfers the kinase, they produce GTP using their kinase activity. Yet, for their function as a mitochondria-promoting protein, they actually conduct it in a kinase-dependent function. So we wonder what exactly how the mechanism that amino promote mitochondrial fusion. So when we take a look at the sequence of amino, we found a very unique sequence that is N-terminus. Within this N-terminus, first, it's unique among others amino superfamily member. Secondly, and it's very conserved from mouse to human. So with this hydrophobic amino acid, we found that this 17 amino acid have this capacity to form aliphatic helix. Aliphatic helix actually has been well-studied secondary structure that can bind to membrane because it's hydrophobic nature from one side and hydrophilic nature from the other side. So we suspect that maybe this N17 region that's unique for amino is critical for their membrane binding and can contribute to mitochondrial targeting and binding. And this is actually the case. When we express simply the N17 amino acid of amino fused with GLP, they can efficiently target to mitochondria in the cell. With the mutation that disrupt their aliphatic helix formation ability, we can see a very dramatic effect on their ability to target mitochondria being reduced. And with that, we speculate is it possible that the amino can bind directly to the lipid, especially phosphoric acid, through its N-terminus amino acid. So we use liposome binding agent, and long story short, we find that indeed the 4-lex amino can bind directly with phosphoric acid containing liposome in N-terminus specific, and also its aliphatic helix formation ability dependent manner. And next, we wonder what if this mitochondrial targeting ability of N7 of amino depends on this PA produced in cell, especially in a PLD6 dependent manner. So we use genetic check that either knock down amino or PLD6 and re-express their counterpart protein and demonstrate that indeed the ability for amino to cause mitochondrial fusion or fission phenotype is downstream of PLD6, suggesting that in cell, amino may bind directly to the substrate of PLD6 to promote mitochondrial fusion. Okay, so next we wonder whether the mitochondrial targeting ability of the N17 amino really depend on the PA generated by PLD6. So either by knockdown of PLD6 or using the optogenetic approach to increase the phosphoric acid amount on mitochondrial membrane, we did see the effect of PA abundance affecting the mitochondrial targeting ability of the N17 region of amino 3. However, when we take a look at the full-length amino, that's a totally different story. That the mitochondrial targeting efficiency of full-length amino is completely unaffected by PLD6. And also, on the other hand, we also express aliphatic helix formation in defect amino, the targeting of the full-length amino mitochondria was perfectly unaffected at all. So we are so desperate. So we decided to go back to what is our hypothetical model. So if you still remember that we mentioned the PLD6 is actually enzyme with its active site facing outward. And therefore, if it catalyzes the production of phosphoric acid, it must occur at 2 contact mitochondria instead of on itself. So with this idea, we think maybe the enrichment of amino or the binding of amino to phosphoric acid should occur at 2 contact mitochondria. So the experiment is easy. We just take a live cell imaging to see where amino localized will be changed during the mitochondrial contact. So this is the case. When we label the amino in yellow, they can see these 2 mitochondria once it contact the signal immediately enhanced compared with the inner membrane labeled mitochondrial marker. And more importantly is that once we knock down PLD6, this kind of contact-induced amino enrichment was significantly reduced, and this in contact induced energy enrichment depends on its ability to form endoplasmic helix, but not its oligomeric ability. So with that, we hypothesize since NH3 is a hexamer and with the binding ability to the phosphoric acid during mitochondrial contact, we wonder if they can function as a tethering protein. To test that, we first use in vitro binding assay. We can see that when we incubate wild type NH3 but not oligomeric defective mutant, we can cluster the first and the oligomer together in a dose-dependent manner, and also using like scattering scattering or TEM, we can see that the NH indeed tether the liposome together instead of promote their fusion. And we also use a hypotonic buffer to stimulate mitochondria in order to see better when different kind of NH is expressed in the cell, whether they will change the tethering ability of mitochondria. And you can see that compared with the control, the top one mitochondrial augmented protein, there is no significant tether ability here. The wild type or kinase dead mutant, they both enhance the contact of mitochondria. So qualified by the contact length measurement. Yet the oligomeric defect mutant or the phosphoric acid binding ability defect cannot induce this mitochondrial tethering. We also collaborate with Yujun in the audience after the sickness fever that we use rapamycin-induced methylation demonstrate that even though the mutant of NH3 that cannot bind to phosphoric acid, as long as we can induce their target mitochondria, they can actually induce the mitochondrial tethering. I hope you can play. Okay, trust me, they were induced the memorization. And also, finally, we want to know what is the function for this NH-mediated mitochondrial tethering. So we think before, when we take a look at the patient skin fibroblasts, when the cell are cultured in the first medium with nutrient fully supplied, that actually the patient cell grow as heavy as the control fibroblast. However, when we culture the patient fibroblast in the glucose-deprived patient, where the mitochondria is heavily dependent on, the cells are first the mitochondria become fragmented, and also the cells start to die. They cannot grow in this kind of glucose-deprived condition. So this data suggests that the intermediate mitochondrial fusion must be enhanced in a nutrient-deprived condition. And actually, back to 2011, the lab, Jennifer Kimko-Schul's lab also published this paper that they found that in the nutrient-deprived condition, they found that mitochondria actually undergo hyperfusion. And this kind of hyperfusion, they propose is because of the phosphorylation of the mitochondrial fission enzyme DRP1. So we wonder maybe there are additional factors that's contributing to this nutrient-deprived induced mitochondrial hyperfusion. So we take a look at NH3, and we found that compared with the control, when cells are cultured in nutrient-deprived condition, the contact-induced energy enrichment is significantly increased upon this kind of nutrient deprivation. So overall, what we want to convince you is that when mitochondria are under stress, especially when the nutrient is not enough, the cardiolipin will be increased and also converted into mitochondrial alpha-lipid. When there are available mitochondrial contact that trigger the conversion of cardiolipin into phosphoric acid, they will promote the selective fusion between start mitochondria. And this control process is dose-dependent. When there are moderate of cardiolipin accumulation, the phospholipolysis can catalyze the phosphoric acid formation to promote selective fusion. However, if there are excess cardiolipin accumulation, of course, it will become a detrimental effect. So overall, this is the work that I would like to share today, and also I would like to thank our collaborators, John, Eugene Lin, and also Jeremy Bosky, and also the talent student Yoan Su. she is the major contributor for this work. And finally, I would say for your attention, and we'll be happy to take your question.
Very nice talk. I actually have two questions. Do you know where is the NME3 location instead of located at the mitochondrial contact site?
Okay. So they say that they actually distribute evenly at the mitochondrial outer membrane.
Also, they are only localized.
Only, I would probably say they also have some cytosolic signal, so they have this equilibrium. When we express it with a atomic expression label, the majority at the mitochondrial outer membrane. But when we take a look at the endogenous label of the NME3, they actually have some cytosolic signal, but the majority still at the mitochondrial outer membrane.
Yes, because that will relate to my second question is, you see that starvation increase the localization of the NME3 at the mitochondrial contact site, right?
Right.
Is that because you increase the protein expression of NME3, or is it because you promote the translocation of the NME3 to the contact site?
That's really nice question. We think that the starvation probably not affect that much of the expression level of NME3. However, I cannot say that for sure if the signal we are looking at here is the ectopic express. So they probably not directly report the amount of the protein expression under starvation. But the data shown here is actually is what you proposed for the second is actually the induce of the enrichment as the contact site. But this kind of enrichment we think is because of cardiolipin. We think cardiolipin will become more externalized to the mitochondrial outer membrane when the nutrient is not known. So we actually are developing probe to have cardiolipin
Also,
for the
promote the enrichment of NME3.
what we speculate that we do not have a cardiolipin probe for now, but that is something that we are working on.
So your liposome fusion is does the NME3 mutant cause the enlargement of the liposome? Because I saw it becomes slightly bigger. So if the enlargement is an independent function of membrane fusion.
I'm sorry for my not explain detail of the data. So you may think this is due to the liposome fusion, therefore we see larger liposome. To be honest, the liposome that we prepare here are we purposely to produce liposome with larger size and also heterogeneous size, because if we produce small liposome with diameter lower than 100 nanometer, the resolution will be it will not be enough for us to distinguish this is one liposome or microliposome. So the larger size here is actually by nature. So here we actually produce small and uniform liposome here, and therefore only the tethering happen, not the fusion happen in the energy crater. So for your question, that we actually don't think that the energy will promote fusion in this context without microfuse. The energy by itself just function as a tethering crater, not fusion energy.
Next up, thank you. You said that under starvation, you externalize the cardiolipin from the inner membrane to the outer membranes. And I got two questions. First, how is cardiolipin externalized to the outer membranes? Is it known? And my second question is that if the cells are under starvation and you recover again the normal growth conditions, so the tubular mitochondria get back to the normal shape?
For the first question, how the cardiolipin is externalized, this have been published a couple of years ago. Where they found that the externalization of cardiolipin at least take two steps. First step is from the inner membrane to the outer membrane, and which is driven by a protein called NDPK4.
That's really kind of funny story. The MTTK4 is actually another member of NME superfamily, and their function was supposedly to fuel as a fueling enzyme to provide GTT for OPA1. And yet it also have a both-side job to function as a cardiolipin exploring enzyme that bind to cardiolipin and transport it from inner membrane to outer membrane. And on the outer membrane, they also need another enzyme called scramblase to flip this membrane to the outer membrane. So you have to take 2 steps for its final exposed to the cytosol protein. And for the second question, it's about how the
during the starvation you have this long tubular mitochondria.
Yes.
And if you recover again the normal condition of the
Will
cells,
become shorter.
Yeah.
Yes, it will. It's a reverse.
is it the fast recovery?
Fast. We haven't did that, but I would examine. I would still probably couple hours work together. I would happy to test that.
Well, I have a question for you related to Manjing's question. So when cell under stress, cardiolipin gets exposed to the outer membrane, is everywhere in the outer membrane or only the contact site? Why yours protein only in the contact site?
Very good question. I also would love to see that. So that's why we are working to develop what can kind of explanation. So according to my students' previous data, it looks like there's no particular spot for the extremisation. Whenever you saw this kind of extremisation, it looks like it's kind of everywhere, but also kind of challenging to increase. Actually, maybe this is the reagent that leads to stress mitochondria is way too high. So I also would like to take this kind of image for the kind of normal condition when the cardiolipin is exposed. I think it should not be everywhere. It should be at the first the contact site between the inner and outer membrane. There's all what they call contact site or where the
so probably there's a response to some kind of lipid signal, some kind of signal, the contact site.
Right, exactly. So I don't think it should be it's everywhere.
Okay, good. Okay, no more questions. Let's conclude this session. And thanks everybody.